Cholesterol is a lipid molecule that helps maintain the structure of cell membranes, and is a precursor to steroid hormones, bile acids, and vitamin D. As it turns out, we make most of our cholesterol ourselves, but some comes through the diet. Cholesterol synthesis, also called the mevalonate pathway, happens in the smooth endoplasmic reticulum of the cell. It starts with two acetyl-CoA molecules getting joined together by the enzyme acetyl-CoA acyltransferase. The result is a 4-carbon molecule called acetoacetyl-CoA, and then a free CoA molecule. Next, the enzyme HMG-CoA synthase combines acetoacetyl-CoA and acetyl-CoA to form a 6-carbon molecule called 3-hydroxyl-3-methylgluteryl-CoA, or HMG-CoA. So 3 acetyls and then a free CoA molecule. Then an enzyme called HMG-CoA reductase reduces HMG-CoA into mevalonate by removing a CoA SH and a water molecule. This step with HMG-CoA reductase is the rate-limiting step of cholesterol synthesis. In other words, the rate of this reaction determines the overall rate of cholesterol synthesis. It's like the slowest step in the assembly line for a factory. Now, cholesterol synthesis is regulated by a trio of proteins: sterol regulatory element binding protein, or SREBP, and 2 others that just go by SCAP and INSIG1. Let's say that cholesterol levels drop because there's less cholesterol coming into the cell from the diet. In that situation, INSIG1 falls off of SREBP, like pulling a pin from a grenade. And the SREBP SCAP complex then gets cleaved by cellular enzymes. The cleaved SREBP floats into the nucleus and binds to the sterol regulatory element on the DNA. When it binds, it increases expression of the genes encoding HMG-CoA reductase. That leads to more HMG-CoA reductase, which speeds up endogenous cholesterol synthesis. Once HMG-CoA reductase has made the 6-carbon mevalonate, it then undergoes a number of additional enzyme-mediated transformations before it becomes cholesterol. First, the enzyme mevalonate 5 kinase uses adenosine triphosphate, or ATP, to phosphorylate mevalonate, creating mevalonate 5-phosphate. Then phosphomevalonate kinase uses another ATP to phosphorylate mevalonate 5-phosphate, making mevalonate pyrophosphate. Finally, mevalonate pyrophosphate decarboxylase removes a carboxyl group from it, forming a 5-carbon molecule called isopentenyl pyrophosphate. Next, geranyltransferase condenses 3 of these isopentenyl pyrophosphate molecules to form a 15-carbon molecule called farnesyl pyrophosphate. Then the enzyme squalene synthase condenses 2 molecules of farnesyl pyrophosphate to form a 30-carbon molecule called squalene. Squalene is pretty cool. It's the last linear precursor to cholesterol, and it's also what helps sharks float. Yep, it helps sharks float.
So I don't sink.
Okay, next, an enzyme called oxidosqualene cyclase converts linear squalene molecule into a structure with rings, a process called cyclization. The result is our first sterol intermediate, called lanosterol. From there are 19 steps of successive modifications, like molding a lump of clay into a beautiful bowl, that convert lanosterol first into 27-carbon 7-dehydrocholesterol, and then finally into 27-carbon cholesterol. Now, it's worth remembering 7-dehydrocholesterol. That's because if it gets hit by a single photon of light in just the right way, then you get cholecalciferol, or vitamin D3, which is important for calcium and phosphate metabolism. Now, with regard to cholesterol, the majority is made and used by the liver, ending up as a bile acid. There are 2 types of bile acids, cholic acids and chenodeoxycholic acids. Both get conjugated by amino acids. Conjugation with taurine makes taurocholic acid, and taurochenodeoxycholic acid, while conjugation with glycine makes glycocholic acid and glycochenodeoxycholic acid. These bile acids are stored in the gallbladder and released into the intestine after meals to assist in fat digestion. From the intestine, some bile acids are eliminated through the feces, but most are reabsorbed by the intestine and get into the portal bloodstream and back to the liver cells. This is called enterohepatic circulation. Now, cholesterol is insoluble in water, so moving through the bloodstream is done through lipoproteins, which are part lipophilic or fat-loving and part hydrophilic or water-loving. Now, aside from the liver, every cell can make cholesterol for itself. One way cells use cholesterol is as a part of the cell membrane. And the role of cholesterol is twofold. At low temperatures, it squeezes in between phospholipid molecules and keeps them from packing too tightly together to keep the membrane more fluid. And at high temperatures, cholesterol pulls phospholipid molecules together, decreasing the space between them. So cholesterol makes the cell membrane fluid and durable, no matter the weather. Finally, cholesterol can be used by the adrenal glands and the gonads to make steroid hormones. Enzymes in the cortex of the adrenal glands tweak the cholesterol molecules to make corticosteroids, like cortisol and aldosterone, which play a role in our fight-or-flight response and regulating blood pressure, respectively. The gonads have a different set of enzymes that metabolize cholesterol a bit differently. The testes mostly use cholesterol to make testosterone, while the ovaries mostly use cholesterol to synthesize estradiol and progesterone. Alright, as a quick recap. Cholesterol is made in the smooth endoplasmic reticulum of cells throughout the body. The rate-limiting step of this reaction is the reduction of HMG-CoA to mevalonate, which is done by HMG-CoA reductase. The molecules are combined to eventually form squalene, the last linear precursor in cholesterol synthesis. Squalene undergoes cyclization to become lanosterol, and lanosterol then undergoes several modifications to become cholesterol. Cholesterol is mostly used in bile acids to help with fat digestion, but it also plays a role in cell membrane fluidity and steroid hormone synthesis.